I'm Samuel by name. I'm a researcher in the Department of Animal Breeding and Genetics, Federal University of Agriculture, Abeokuta, Ogun State Welfare Group, Nigeria. And this group was funded after a meeting of students and lecturers in 2019. So what is our mission? Our mission is to increase awareness of animal behavior and welfare in Nigeria and Africa at large, and to foster collaboration and networking among researchers who are into the field of animal behavior and welfare. And for these our presentations, people have presented on animal welfare issues such as fear, ethical practices. Some people have talked about cognition, while some people have talked about stress. Today, Tom will be talking about stress. And today's presentation is the 13th in the series. We have presentation in October, November, and December. So we have 6. We have done 13. So we'll be doing the 13th today. So the moderator for today's presentation is Dr. Oluwaseun from the Department of Animal Physiology, Federal University of Agriculture, Abeokuta, Ogun State, Nigeria. And today's presenter is Dr. Tom Smulders from Newcastle University, and he will be talking about neural markers of chronic stress in poultry species. I will hand you over to the moderator to give us the rules. Dr. Yasiri.
Good afternoon, everybody from Nigeria, and welcome to today's seminar presentation series. I'm Dr. Oluwaseun Yasiri, like the host said, and I'm a senior lecturer in the Department of Animal Physiology, Federal University of Agriculture, Abeokuta. Dr. Tom obtained his undergraduate degree in zoology from Antwerp University, Belgium, and his PhD from Cornell University, USA. Studying the role of hippocampal formation in spatial memory in food hoarding birds. He has continued to study the avian hippocampal formation ever since he started as a lecturer in Newcastle University in the UK in 2002. In the last 10 to 15 years, he has started looking at the avian hippocampal formation as an indicator of chronic stress in birds, and has used this to assess the long-term welfare state of chickens under different conditions. So today we have Dr. Tom, and he'll be presenting on neuromarkers for chronic stress in poultry. Before
the invitation, I'm very excited to be presenting to this very broad and international audience. I'm going to tell you a bit today about how we've been using neural markers of chronic stress in our pursuit to try and measure welfare in chickens, and how that might potentially bring something new, a new tool to the welfare toolbox. Now, as you all know, if we're looking to improve welfare of farmed animals anywhere, chickens are a good place to look, because there are every year roughly 26 billion chickens being farmed around the world. At least that's what Google tells me, because I just looked it up. And of course, how these animals are being held varies very widely, from backyard chickens to chickens in laying batteries, etc., etc. Now, in the West, at least in Europe and the United States, there's a trend away from cages, at least from traditional cages, to either bigger cages or cage-free systems. And there's still a lot of questions about what is good and what is bad about the different alternative systems. I think everybody agrees that the traditional battery cages are very poor for the animal's welfare, but which of the other systems is better or worse is still very much a question of research, really. And one of the things that we think is important is, of course, to figure out what's good for their welfare. Now, welfare can be measured in many different ways. I'm thinking here about the aspect of welfare that relates to the long-term experience of an individual. So what is the chronic experience? Is it overall good? Is it overall bad? And, is it a sequence of some bad things with very much neutral, or some bad and some good? And so a measure of welfare, ideally, would be something that integrates over a long period of time. and that is sensitive to whether the experiences were good or bad. And one of the things that will happen with long-term negative experiences, or many negative experiences, is that the animals experience something we call chronic stress. So essentially that their stress response, the physiological systems that are, that evolution has put into all vertebrates to deal with unexpected circumstances to make sure the animal survives in the longer term. But those systems, when chronically activated, tend to be associated, definitely in humans, with chronic negative affective states. So we experience them as chronic negative states. And so if we can measure something that relates to those chronic negative states, especially that tells us something about the longer term experience of the animals, then that might be a good marker to tell whether the animal's welfare is poor or better. Now, we have inspired ourselves on the literature, the vast literature on preclinical studies in psychiatry. So this is the studies that use rats and mice as model systems for studying humans, really. And they're very often used for example, developing new antidepressant drugs, or indeed many other ways of trying to understand how the brains of people with depression, and by extension rats and mice with depression, are different from those without, and if we can, what kind of treatments can improve that. Now, rat and mouse models of depression are very often induced by exposing the rats and mice to chronic stress. And so now we are, in the same territory that I was just talking about for animal welfare. We have here a system where chronic stress is a treatment that's being done very commonly in the literature, and one of the common consequences of these chronic stress treatments, and it doesn't even matter which kind of stressor is being used, it's all of the chronic stressors that lead to depression-like symptoms usually also lead to a reduction in a process in the brain called adult hippocampal neurogenesis. So that's what I'm going to first introduce to you. What is adult hippocampal neurogenesis? And then I'm going to show you how we have used this process to look for chronic stress in chickens under different conditions and different circumstances. Okay, so the first thing I want to introduce to you briefly is a mouse brain, and not only because that's where most of the literature, of course, is. This is a semi-transparent model of a mouse brain. Inside of it is the hippocampus, which is this pink structure. And importantly for this discussion, we can divide that into two sides, the septal pole on the one hand and the temporal pole on the other hand. And we know from a large part of the literature that especially the temporal pole of the hippocampus is especially sensitive to chronic stress. Septal pole can be as well, but the temporal pole is more reliably so. We also know that the temporal pole is more involved in regulating the stress response and regulating emotions, whereas the septal pole is more involved in cognition and memory. Now, adult hippocampal neurogenesis is a process through which new neurons, new brain cells are being born constantly throughout life. And that happens in a mammalian brain only in a few restricted parts of the brain. And one of those parts of the brain is the dentate gyrus of the hippocampus. So if we take a cross section here of this pink banana shape, we will see a thick layer of cell bodies, which kind of ends in the middle of another arrow-shaped layer of cell bodies. And it's in the middle of this arrow-shaped layer of cell bodies called the dentate gyrus that we find adult hippocampal neurogenesis. Specifically, it's a one-cell layer thick zone called the subgranular zone, which is blown up here. This subgranular zone sitting here at the bottom contains these stem cells, which from time to time will divide and basically make a new daughter cell. That daughter cell will then divide several times. It'll be a type 2 cell, an amplifying cell, divide several times to get multiple daughter cells. And those daughter cells will then differentiate into new neurons by growing out dendrites and axons. And these immature growing neurons express a protein as part of this growth called double cortin. And that's important because we're going to use this protein as a marker of neurons that are immature, that are recently generated. Okay, now It is this process from stem cell division through to maturation that is affected by chronic stress in all these rats and mouse models. And so here's just one example study of this, and paper from 2010, but there's many, many, many examples. In this case, the stress was chronic restrained stress. So the mice were restrained for 6 hours a day for 4 weeks straight. I think they might have had Sunday off. So that means 6 hours restrained a day, which is a stressor for these animals. And so looking here, we see again this arrow shape, the dentate gyrus, and we see the subgranular zone, and we see it in a control mouse here and in a stressed mouse here. Now, these are 3 different measures of neurogenesis. Ki67 is a marker of mitosis. So any cells that are undergoing cell division at that moment in time when the animal was killed will express Ki67. And so we can stain the cells so that we can see all the cells that express Ki67 as black dots. And we see there's more of these black dots in the control animals than in the stressed animals. BRDU is another marker. This is one that you have to inject. And when it's injected, it'll incorporate into the DNA of any cells in the body, really, that are in S phase of mitosis. So it's synthesizing new DNA. Now, if you inject the BRDU before the stress treatment, but only kill the animal after the stress treatment, what you're basically looking at is cells that have survived for the whole period of the stress treatment, but that were born right before the stress treatment when the BRDU was injected. And again, what we see here is that more of these surviving cells are found in the control animals than in the stressed animals. And finally is our marker double cortin that we're going to be using. This is a protein expressed by the maturing neurons. And that's why we don't just see it in the cell bodies, but we also see it in all these dendrites. But again, we see more black here in the control animals than in the stressed animals. So we're going to use this same process, all using double cortin in this case, to look and see if the animals that we're comparing to each other have more or less of the double cortin, and less of the double cortin will therefore be interpreted as having experienced more chronic stress, because that's, at least in rats and mice, commonly, is a reliable marker. Now, you should know that in rats and mice, just treating animals with exogenous corticosterone also reduces adult hippocampal neurogenesis. And of course, we all know that corticosterone is released as part of the stress response. Now, importantly, corticosterone is increased as part of the stress response, but not only to negative experiences, because our stress response is involved in any kind of preparation of the body to deal with a change in the environment. And so corticosterone is also increased, for example, when animals get access to sexual partners. It's increased when animals get access to novel environments which they need to explore. It's increased when animals get access to food, maybe after they haven't had food for a while. So there's many positive experiences where corticosterone is also increased. So if hippocampal neurogenesis just reduces whenever corticosterone is high, then it wouldn't be a very useful marker, because corticosterone is a marker of arousal, not of valence. It goes with positive and negative experiences. But luckily, as you might expect, that is not the case. It doesn't just follow corticosterone. So there was a paper published in 2013 which showed this very clearly. And so I'll briefly take you through the results just to show you. In the first experiment, what they did was they had 2 groups of mice, one just housed in a home cage and one with chronic stress, 2 weeks of social defeat. And half the mice were intact, and half the mice had their adrenal gland removed and just had a steady implant of corticosterone, but the corticosterone could not change with the experiences. Now, what you see, as you might expect, is that the social defeat mice, the sham ones, the intact animals, had less neurogenesis than the home cage mice. Yeah, that's what I've just been saying. Chronic stress reduces in this case is measured as BRDU. So fewer new neurons in the stressed animals than in the control animals. But if you take the adrenal gland out, they have more neurons, right? So that says indeed corticosterone is responsible for this reduction, and without corticosterone, if anything, we even get an increase in new neurons. That's fine. That's what I've already been saying. But what they also did was they then did another experiment in which the treatment was followed by the surgery, and then they were all given enrichment. Now, the enrichment also increases corticosterone in these sham animals, in the intact animals, of course, not in the adrenal ectomized animals, because they don't have an adrenal gland. But what we see there is that enrichment increases neurogenesis, as has been shown many times in the literature. It increases neurogenesis, but only in the intact animals. If the adrenal gland cannot release extra corticosterone, the increase in neurogenesis is also not possible. So that tells us that even though corticosterone itself may always go up, whether it's to a positive or negative experience, neurogenesis follows the valence of the experience. So corticosterone increases both in negative and positive conditions, but adult hippocampal neurogenesis decreases when it's a negative experience, but increases when it's a positive experience. And that's important because otherwise it wouldn't be a very useful measure. It would be much easier to measure corticosterone itself. But it isn't just a readout of the corticosterone experiences. It is a readout of the valence of the experiences as well. Good. So that's background. Now, what about birds? So this was all about rats and mice. And you might say, well, all good and well for rats and mice, but is the same thing happening in birds? And of course, bird brains are very different from rat and mouse brains. Here's a chicken brain up here. Here's our diagrammatic mouse brain down here again. And in a chicken, the hippocampus is not sitting inside the brain looking like a little sausage or a little banana. It is in fact sitting on the surface of the brain in the back of the brain. So this is the back, this is the front. We're looking at the top down on the brain. Hippocampus sits here at the back of the hemispheres on the surface, okay? That's okay. The fact that the position is different, of course, doesn't matter. Our question is there adult hippocampal neurogenesis and is it sensitive to chronic stress? And so that's what we're going to test first of all. When you do cross sections through this brain, so cut sections like this, the hippocampus is this structure that starts in the middle, in the front, and then you see it kind of wrapping around the top as you go further back because it sits here, and then all the way around the back of the brain. Now, remember I told you that the temporal pole in mice is more sensitive to stress than the septal pole. We hypothesize, and some of the evidence seems to suggest that we might not be wrong, is that the back of the hippocampus, the very back of the chicken hippocampus, is equivalent to this temporal pole. And so we will specifically be looking separately at the caudal part of the chicken hippocampus compared to the rostral part, because we expect the caudal part to be most sensitive to chronic stress. Now we stain these sections with an antibody against the double coating protein, which I've already told you about. And we count cells that have two different morphologies. One is multipolar cells. So here we have a cell that has, you can probably just see processes coming out of this cell. So it has more than two processes coming out. So we call it multipolar. The other types of cells that we count are bipolar. So they just have two processes, front and the back. Now we interpret bipolar neurons as possibly migrating. So having been born in one place, but going to another part of the hippocampus. Whereas multipolar cells are interpreted as more mature and settling and making connections in the hippocampal circuitry. So we count them separately because we believe they might represent, or we're not sure of this, they might represent different developmental stages of, or different differentiation stages of these neurons. Right. So our first question, of course, is, does chronic stress actually reduce hippocampal neurogenesis in birds? Because just because it does in rats and mice doesn't mean it has to in birds as well. Birds and mammals have, had separate evolutionary histories of more than 300 million years. It is completely possible that even though both have a hippocampus, that the hippocampus in birds doesn't respond to stress, or at least that adult neurogenesis doesn't respond to stress. So the first experiment we did, and this was with Fabio Gualtieri here and George Longmore, who did this experiment. We exposed brown-laying hens to 8 weeks of chronic mild stress. So what they experienced was a series of stressors, every day a different stressor, never knowing which one was coming the next day. And so the stressors included being isolated in a box for an hour, having a fan in their pens, because apparently, and this is what my colleagues that know more about chickens tell me, chickens do not like wind. So we put a fan in there. It's also loud as well, so there's noise involved too. Or we took their food away for a few hours. Sometimes we sprayed the whole pen and the chickens with water in the evening and left it wet overnight, which also increases the smell of ammonia in the room. Sometimes we increased the temperature for a few hours, or we decreased the temperature for a few hours. And we also changed the day length. So for one thing, there was an unpredictable light-dark cycle going on. And on a few days, we increased the days to 21 hours of light, and some days we increased it to 21 hours of darkness. So basically, they never knew what was coming. And that's a protocol that in mice, with slightly different stressors, because mice and chickens don't necessarily find the same thing stressful, is very, very efficient at reducing adult neurogenesis in the hippocampus. And indeed, we found the same in chickens. So when you look at how much neurogenesis we find in the caudal and the rostral hippocampus, these are the control animals which had none of these stressors. These are the stressed animals. The counts have been Z-scored, so they're, the mean is 0 and the standard deviation is 1, which is why we get some negative values. But what we see is there's very clearly in the caudal hippocampus a very clear difference in the density of, in this case, multipolar double cortin positive cells, just like we would predict from the mouse literature. So many more double cortin positive new neurons in the control animals than in the stressed animals. And it's specifically strong in the caudal part of the hippocampus, which we were predicting as being equivalent to the temporal pole of the mouse hippocampus. The difference is in the same direction in the rostral one, but it's much smaller and it's not significant. Okay, so we can conclude then that adult hippocampal neurogenesis is indeed reduced by chronic stress. That is stronger in the caudal hippocampus, so we definitely need to look there if we're going to look for effects of stress. And that means that we can now use this marker, the level of adult hippocampal neurogenesis, or the density of double cortin positive neurons, more specifically, as a marker of chronic stress. And we're going to now apply that to several more commercially relevant questions for laying hens. So the first one that we looked at was keel bone fractures. Now the move from battery cages to non-cage systems has also, as a consequence, had a large increase in the occurrence of keel bone fractures in hens. The keel bone, of course, is the sternum, the big bone at the front that the flight muscles, the breast muscles are attached to. And when birds are living in these multi-tier environments, they, of course, have to go up and down. And sometimes when they fly down, they will miss a perch, or they will land heavily with the chest onto the perch, maybe because another bird got in the way, maybe because they misjudged their jump, etc., etc. But it is very common for birds to have keel bone fractures. And whereas I don't think anybody disagrees that the actual fracture and healing from the fracture is probably painful, we do not know whether there are chronic effects of having keel bone fractures, or if it's basically a question of healing and then they'll be fine. So what we did was we collaborated with a group in Switzerland, Mike Toscano's group, and they were doing a long-term study on the development of keel bone fractures. So they were taking x-rays 11 times during the 21 to 60-week-old age range of these animals. So they followed the same animals from 21 weeks old to 60 weeks old. And every so many weeks, they would take the focal animals out, take an x-ray to see if they had keel bone damage, and also so they could follow the same individual over time and see how their keel bone damage developed. And we used an aggregate score of fracture severity at the end of life to then relate to the adult hippocampal neurogenesis that we measured in our lab. And this was work done by Elena Armstrong. The animals were also tracked, but I'm not going to present any of the behavioral data here. But Mike's group tracked which part of the aviary they were actually using as individuals. So the focal animals were carrying a little logger that got information from these little, I think they're infrared senders on each of the tiers of the aviary. Okay, so what Elena found, so I'll point you to what you're looking at. We're looking again at these Z-scored cell densities. So again, positive and negative numbers because the mean overall is 0. In white is a group of animals that had minimal keel bone damage. It's almost impossible by 60 weeks of age to find ones that had no keel bone damage at all, but we took the ones that had the least keel bone damage, and it is very minimal. Whereas the black bars are animals with severe keel bone fractures. Here we're looking at multipolar neurons. Here we're looking at bipolar neurons. And then in each, we're looking at rostral and caudal hippocampus separately. And the statistics are below here. And both for multipolar and bipolar, we see a significant effect of keel bone fracture. So more double cortin positive neurons, both in rostral and caudal this time, in the minimal group, and many fewer in the group with severe keel bone fractures. For the multipolar neurons, there was even a significant interaction between group and subregion. So even though both rostral and caudal showed significant differences, the difference was significantly larger in the caudal. So again, the part of the brain, the part of the hippocampus we believe to be the more sensitive, to stress, shows us the bigger difference between the control animals, if you want to think of them that way, the minimal keel bone fracture animals, and the animals that had severe keel bone fractures. But given the longitudinal data that we had, we could actually go further and look at something else as well. We could look at how long ago each animal had acquired its first keel bone fracture. And what we found, so again, we're looking at the density here, multipolar neurons, bipolar neurons, but the two graphs are very similar for both multipolar and bipolar. The gray dots are the caudal hippocampus, the sensitive part, and the black dots are the rostral hippocampus, the not so sensitive part. And we find that indeed in the caudal, but not in the rostral hippocampus, we find a correlation, a significant correlation between how long ago the first break happened and how many double cortin positive neurons they have in their caudal hippocampus. The ones that had their breaks very early on have fewer neurons than the ones that had their first break more recently. And so that suggests that there is definitely a chronic effect of having keel bone damage. So it's not that these animals had their break, 40 weeks ago and they're now fine, they're healed. No, it looks like that break will have had continuous chronic effects on the animal's welfare. And therefore their hippocampal neurogenesis, now that they're 60 weeks old, is lower than animals that have been able to live for longer without keel bone breaks and then broke them, say, 18 weeks ago or 15 weeks ago, before they were killed. So that suggests that indeed keel bone fractures do reduce adult hippocampal neurogenesis, and the more severe the keel bone fracture, the more it reduces the adult hippocampal neurogenesis. I didn't show you that, but we had some evidence on individual differences there as well. But more importantly, the longer they live with the keel bone fracture, the more the hippocampal neurogenesis, at least in the caudal hippocampus, is being reduced, which suggests that living with existing and even healed keel bone fracture is a chronically stressful, chronically stressful thing to do. And we would even call it a depression-like state because it's equivalent to what we see in these mouse models of depression, which also see this reduction in adult hippocampal neurogenesis. So that suggests that obviously reducing the chances of your birds breaking their keel bones isn't just important because actually animals without keel bone damage lay more eggs, because of course bone damage requires calcium to repair, which then takes calcium away from eggshells. So, I think farmers know that keel bone damage isn't good, but this actually shows us that it isn't just not good at the time of the fracture, it is not good for the rest of the animal's life and causes quite a strong chronic stress throughout life. What about more normal, so to speak, housing system comparisons? We would in this study, Elena again, compared animals housed in a typical British free-range egg situation, which means indoors they're in a tiered aviary, but then they have these pop holes so they can go outside to forage outside during the day. they can go, they're not forced to go outside. The pop holes just open up and then the birds can come out if they want to. We compare that to another fairly common system in Britain, which are these large enriched cages. So these are not battery cages. These are large cages where 60 birds live together in a cage, and the cage has different compartments. It has a laying compartment, a scratching compartment, food and water compartments, etc. Some of them separated by some curtains. So the birds have structure within their environment, but it's all indoors and it's smaller in space than these animals have. These animals live 4 to really 16,000 in a flock. These animals live in social groups of 60. So, one might argue that maybe a social group of 60 is less stressful than a group of 4,000 or 16,000. On the other hand, these animals have much less space. They don't have fresh air. They don't have outside light, etc., etc. So might there be differences in chronic stress between these two housing conditions? Now, in order to test that, we sampled from the two housing conditions. And because we know that there are individual differences within a housing system, we thought we would sample from the extremes of physical condition within each of the housing systems. So in each housing system, we sampled 12 what we called good birds. And these are birds with higher feather cover, higher body weight, and redder combs and wattles. And we also sampled 12 poor condition birds, which had lower body weight, lower feather cover, and very pink to pale combs and wattles. In the multi-tier free range, we took the good birds all from outside, so we also were sure that these birds used the outside area. And we took the poor birds from the top tier inside near the food and water, which made it more likely that these are birds that don't really go far away from their main resources, probably because they're not able to do so. But we didn't track individual birds, so it's very possible that another time of day these birds might have gone outside. We do not know that. We had a slight confounding problem with this particular study, that our birds were actually from different strains for the two different systems. So the free range birds were an H&N bird, and the colony birds were highline bird. But we controlled for this by collecting pullets of those two same strains, that are, before they moved to the laying environment. And we found no differences in adult hippocampal neurogenesis between the two strains. So we assume that any differences we do or do not see are due to the housing system rather than to the background strain of the animals. So what did we find? Again, we're getting used to these graphs now, I hope. We have the Z-scored cell density over here. In this case, we have rostral over here, caudal over here. We're just looking at multipolar cells for this particular graph. And within it, we've got our good condition birds in red, poor condition birds in gray. Dark ones are the free range multi-tier system. Light ones are the cage system. And the main conclusion is that we don't find an effect of housing system itself, but we do find an effect of condition. So poor condition birds, whether they from the colony, from the cages or from the free range, have lower amounts of neurogenesis, both in the caudal part of the hippocampus and in the rostral part of the hippocampus. And it doesn't matter what housing system they come from. So the conclusion for this is that actually housing system per se does not seem to reduce adult hippocampal neurogenesis, whether you go to cages or you go to these free range systems, as long as you compare birds of similar physical and presumably similar mental condition in the two systems. Because we do find that animals in poor physical condition have lower hippocampal neurogenesis, therefore presumably experience more chronic stress than birds in good physical condition. So what this tells us, and maybe this is no news to people who actually do work in housing systems, that maybe the relevant comparison between housing systems should be how many high quality, how many high physical condition, good physical condition birds are there, and how many poor physical condition birds there are in any housing system. So a housing system with a higher proportion of low physical quality birds or low physical condition birds is, per definition, a worse welfare system, because that has more of these chronically stressed birds in it. And we don't know which one that is because we did not go through the cages and sampled systematically to see how many of these good and poor condition birds there are. But that would be a very interesting study to do next, because I could see it predicted both ways. I really don't know which way that would come out between the free range system on the one hand and the cage system on the other hand. The final study I'd like to tell you about has to do with another physical condition that the birds experience quite commonly, and that is footpad dermatitis. So this is the first study of Matt Craven's PhD, we can see there in the corner. This is a collaboration with the group of Frank Tuitens in Belgium. Now they were just about to get rid of a flock of birds, and they knew that there was variability in the footpad dermatitis within their flock because they had been scoring footpad dermatitis in their flock. And so we came along and collected brains at the end of their study. And we looked for birds that had either a lot of footpad dermatitis or very little footpad dermatitis. So footpad dermatitis was scored as either 0, none, 1, which was some damage, some necrosis on the foot pad, or 2, which would be necrosis and swelling, where the swelling could be kind of seen from the bottom. So the top of the foot swells, so it bulges over at the bottom of the foot. And so they were scored on 3 occasions, in May, in July, and in September. Now we came along early October, and we picked birds for the no footpad dermatitis that had 0 scores, ideally on all 3 occasions, maybe 1 on 1 occasion. That was the maximum that we'd allow. Whereas then we also looked for birds that had 2 on all 3 occasions, or as close to that as possible for the high footpad dermatitis birds. And we compared these to each other. Now it turned out that we had another complication in this sample that I'll explain to you in a minute. So let's look at these birds that here first. So this is the cumulative footpad dermatitis score, so maximum 6, which is 2 on all 3 occasions, or 0, which is 0 on all 3 occasions, and everything in between. And this is bipolar cell density and multipolar cell density. And again, we see that there's a negative relationship. So animals with more footpad dermatitis damage had fewer bipolar cells and fewer multipolar cells. Red is the caudal hippocampus, blue is the rostral hippocampus. It happens in both parts of the hippocampus again. So that's confirming what we predicted, that the stress of footpad dermatitis, which is presumably painful, also reduces adult hippocampal neurogenesis, which we then would interpret as that means it is a chronic stressor. It isn't just something that the birds can ignore. It is chronically stressing the animals, like any chronic pain would do, presumably like the keel bone damage. But notice here, these are birds that had been raised in the normal, traditional commercial way, which means hatchery, then straight to the rearing facility, normal open floored rearing facility. It happened, however, that Frank Tuitens in that particular study had 2 groups of birds mixed together. These are the birds that had been reared normally, commercially normally. He also had a group of birds that had been reared with dark brooders. So dark brooders are these warm, dark places, allowing the chicks in the early stages after hatching to hide under, to look for warmth and to look for darkness. They are meant to mimic the wings of the mother hen that the birds can hide under when they feel unsafe or when they feel cold. Now, the birds that had been reared with dark brooders, remember that these birds are all old birds. These are adult end-of-lay birds. There, we also had variability in footpad dermatitis, but no relationship, if anything, slightly increasing, but actually that's not significant. So basically no relationship between footpad dermatitis and hippocampal neurogenesis. So it looks like this very early life experience in the first few weeks of life of having this dark brooder available made the birds more resilient to this reduction in neurogenesis due to the footpad dermatitis. So the birds were not experiencing it as much as chronic stress if they had had this early life experience, which presumably therefore set up their stress system to be more resilient to such chronic stressors. But this is a very early results. We weren't expecting this, we weren't predicting this. We actually had forgotten all about the fact that there were birds with dark brooder experience and ones without until we analyzed these data. So in conclusion for that one, so footbat dermatitis, like keel bone damage, does reduce adult hippocampal neurogenesis, and the more the worse it is, the more it reduces it. But it seems to be that early life experience with dark brooders may make birds more resilient to these kinds of stress. And so we really want to look a bit more into what dark brooders actually do to the stress resilience and the stress responses of birds throughout life. So in conclusion, we are looking at these double cortin positive neurons in the hippocampus, which seem to be sensitive to chronic stress, and therefore we can use them as a marker of whether chickens have experienced chronic stress under certain conditions, compare them to animals that do not experience those conditions, and then we can say, well, that condition seems to be more stressful than that condition. And so we have been using this in the context of keel bone damage, of physical condition, which might indicate quite a lot of different types of stressors, and in the context of housing systems, as well as now these new results on footbat dermatitis and the intriguing results of possibly different developmental programming going on early on in life, dependent on whether or not they have access to these dark brooders. So I'd just like to conclude by thanking all my collaborators, Tim Boswell, who's working with me on all these projects. This is Mike Toscano, whose Swiss birds we used. I'll point out, I think, yep, that's most of the people. I really need a picture of Frank Tartans here to thank him as well. And these are the students that did all the work down here. So I will leave it to that. I'll stop sharing my screen for now, turn my camera back on, and then Seon can tell me if there's anything I should be answering.
Yeah, thank you very much, Tom. That was quite an interesting presentation on neural markers. And I'm so glad that at least you've done a lot as regarding welfare issues, relating the use of the adult hippocampal neurogenesis on footbat dermatitis and chronic stress, an indicator of pain. Also, I got interested when you got to the point about talking about the use of the dark brooders as a means of simulating the maternal care. And I'm so glad about that result. And I'm hoping that we would really know how that had really helped to reduce, how the birds were able to experience or go through life not experiencing much pain as those that never had the dark brooders. But
yeah, I'd like to know too.
Yeah. So you talked about the use of the chronic mild stress paradigm for one of the experiments, but I was just wondering if the effect of the use of a chronic mild stress paradigm might be similar to the use of a chronic but predictable stressor type of protocol. What do you think about it?
In chickens, we don't know. But in the rodent literature, it is clear that the unpredictability is part of what makes it so stressful. So a more predictable chronic stressor is less stressful, so reduces adult hippocampal neurogenesis, is less likely to induce depression-like states in rats and mice. So predictability generally is part of what makes stress more manageable. But we haven't done the experiments in the chickens, so I can't tell you exactly by how much less it would affect adult hippocampal neurogenesis. But generally, the stress literature suggests that predictability helps, essentially.
We have a whole lot of questions in the chat box for you, and I'll try to, let's see how far we can go. Dr. Duroshara is asking, what other parts of the brain can be used to assess chronic stress apart from the use of the hippocampus?
Well, the honest answer is I don't know. We've been focusing on the hippocampus because of the large literature on hippocampus in rodents, and the fact, very practically, that that is the part of the brain in birds that I've been studying since my PhD, so I know it quite well. There are obviously parts of the brain, other parts of the brain involved in regulating the stress response. But what's so special about the adult hippocampal neurogenesis specifically is that we know it responds specifically to long-term chronic stress and kind of can leave a record of that. Now, it is not unreversible because we know that in rodents, if you take the stress away and you put the animals in an enriched environment, it can go up again. So it's not like you have a permanent record. It can be reversed, but at least it is something that, you know, is a somewhat longer term record. And I don't know of any other parts of the brain where that would be also the case. That doesn't mean they're not there. I just don't know what they might be.
Okay. Raleigh wants to know how adult hippocampal neurogenesis, can it differentiate between the absence of chronic stress and the presence of positively valence experience?
So I think that whatever you go, so this method is only really usable relative, like by comparing two groups to each other. You cannot just take one group, measure adult hippocampal neurogenesis and say, okay, they are at this level of welfare or that level of welfare. It's always relative to each other. And so when it is relative to each other, I don't think I could tell the difference between neutral and positive on the one hand, or slightly negative and neutral on the other hand. You know, the difference in adult hippocampal neurogenesis between those two sets might be exactly the same. So I think all you can do with this is take two and say which is better than the other. And even then, I think the differences have to be big enough because I don't know how sensitive this measure is. It clearly works for things like keel bone damage. It clearly works for things like being in a poor physical condition for a long time. But I do not know if it would work for, you know, I have one mealworm extra a day over that group or something like that. So, and because it takes so long to do all these measurements, it's very hard to try and start titrating all that, to try and find exactly how sensitive is it, exactly how long does the stress need to last, because that's many, many studies, many, many brains to process, and therefore many, many years worth of work or person years worth of work. Which is why there's a load of good questions that I really don't know the answer to. I would love to know the answer to. But generally, it does seem to be that positively valenced experiences do increase hippocampal neurogenesis in definitely in rodents. We haven't done it in birds yet. And so therefore you should see similar things going from negative to neutral as you do from neutral to positive.
So an increase in the adult hippocampal neurogenesis could be an indicator of positive valence?
More positive, sorry, that was my phone. More positive than the other group. So that's all, you know, it's always, so it's either more positive or less negative. And I don't think the measure could tell whether it's more positive or less negative. It's going in the right direction. And that's about all I think it could say.
Thank you. Another question, do you know if the relationship between adult hippocampal neurogenesis and chronic stress is linear, or might it be an inverted U-shaped relationship?
Yeah, it might be inverted U-shaped relationship. So, you know, I told you that increases in corticosterone can both be involved in increasing and in decreasing adult hippocampal neurogenesis. What I don't know is if those increases are equivalent. So at least in rats and mice, it might be that the increases that lead to increase in neurogenesis might be slightly smaller than the increases that lead to decreases in neurogenesis, if I'm not confusing people with all these increases and decreases yet. Personally, I don't think it's just a question of the level of corticosterone. I think it's a question of corticosterone interacting with other signals that tell the hippocampus whether it's a positive or a negative experience. Things like inflammation or, you know, other aspects of chronic stress that distinguish between why cort is high. But again, to do a relationship like that, to plot that curve would take a lot of animals. And I don't think even in the rodent literature, people have really done this yet. And they, of course, can use a lot more animals with mice. And mice are definitely a bit quicker to use than chickens. Their brains are slightly smaller too, so it takes less time to process. Still takes a while.
Thank you, Tom.
Anyway, sorry.
Dr. Duro Sharo wants to know, what is the relationship between keel bone fracture and cognition in chickens?
No idea. But if adult hippocampal neurogenesis is involved in cognition, which many people believe it is. The keel bone fracture did reduce it also in the rostral hippocampus, which is hypothesized to be more involved in cognition than, oh, sorry, did somebody turn me off on purpose?
No.
Okay, so so I would think that it probably would affect cognition, but I don't know to what degree and which aspect of cognition you'd need to measure. So I don't know if you could, I mean, it would be nice if you could get a behavioral readout of hippocampal neurogenesis, wouldn't it? You could say this cognitive task tells us whether there's more or fewer of these adult hippocampal neurogenesis neurons. In rodents, there are some tasks that people claim are sensitive to the amount of neurogenesis going on, but definitely in birds, nobody's even tried it. Not in chickens, at least.
Abba from Sudan, he wants to know, what is your plan or opinion on how to reduce chronic stress now that we know that it's a big issue in how can we reduce it?
Yeah, it's a big question, isn't it? So this is where, you know, I feel I'm the person that's trying to work out some measurements. That's why I collaborate with people closer to the industry and closer to poultry science to kind of see, okay, they need to think about what they can improve, and then we could use this measure to see if it actually did improve life, or is it actually worth doing. So, you know, from the last experiment, maybe dark brooders are a useful step. Some people, of course, are saying that we should stop intensive farming altogether because it's the intensive farming that's the problem. People say we should abandon cages. I am not so sure that the big cages are necessarily worse than the living in a barn with 4,000 animals completely packed together. To me, they're open questions still. And I definitely am not designing new housing systems, but I'm very much open to collaborating with people who have ideas of something that might reduce chronic stress. And we can use the brain measure to see if it really does reduce chronic stress.
Thank you. What is the relationship between fear and adult hippocampal neurogenesis in poultry? Is there any relationship?
I don't know. I could only speculate that regularly being exposed to fear, regularly being in a fearful state would be a chronically stressful condition. If it's an acute fear, I doubt that it would do much if it's only once or twice. Whether a chronically stressed animal is more easily afraid, that might be the case, because anxiety is one of those things that in rats and mice also goes hand in hand with decreased adult hippocampal neurogenesis. So it is possible that more chronically stressed animals are also more anxious and therefore more fearful to different situations. I would think in that case, it's probably the fear is a consequence of the reduction in adult hippocampal neurogenesis rather than a cause of it. But I mean, that's just speculation, really.
Yeah. So Raleigh wants to know that, is it possible that the dark brooders don't show reduced adult hippocampal neurogenesis to dermatitis because their developmental experience reduced their physiological, the HPA axis response to stress? Could that be the reason?
Yeah, could be. I mean, as I said, we found this result, it was unexpected, that we know there's a lot of literature that shows that early life experiences can program the HPA axis in different ways. And I wouldn't be surprised if that was part of what was happening here. And then the question becomes, is having a less responsive HPA axis also associated with experiencing these things as less stressful? Or is it just that one aspect of the stress response is not working, but the animals are suffering just as much as the other animals? And that, of course, we can't tell, right? Not from this. That would take a whole series of experiments to try and pull those things apart. But yes, absolutely possible.
Thank you. Surya from Malaysia is asking, is any stress-related genes isolated? Can any stress-related gene be obtained from the hippocampus?
Yeah, no, Surya, it's a good question. We have been trying for a while to use qPCR in the hippocampus to try and get a quicker way of measuring, the consequences of chronic stress. Because this, as I said several times, staining these neurons, counting them, it is a very slow process and it takes a long time. We haven't had any luck yet to get anything that's robust and reliable. So we've given up now on double cortin as a marker, because it turns out that the mRNA for double cortin is probably around much more than just in the cells that express it enough to be counted. So the mRNA from the whole hippocampus does not seem to change along with the numbers that we count. We're looking at mRNA for BDNF, brain-derived neurotrophic factor, which we know is also involved in neurogenesis. Again, so we're using this, we're testing these in the same brains that we've gotten these clear results on the anatomy, because we took the other hemisphere and we extracted RNA from the hippocampus in the other hemisphere. So we know that we should find differences in these birds because we've seen them in the anatomy. And so far, BDNF hasn't worked, double cortin hasn't worked. Other markers to do with cell division don't seem to show any differences. So our next process that we're going for is to look for inflammation markers, especially markers to do with microglia, to see if brain inflammation might be a marker of chronic stress in the hippocampus. But so far, no luck there either, but we're not done with that quite yet.
Thank you, Dr. Tom.
You're welcome.
So I think well, like all participants, you can unmute and give few comments before we end. We have just 2 minutes to that. You can unmute, you can turn on your video. We really appreciate Dr. Tom Smulders for this wonderful presentation, and I've really learned a lot. But I just want to ask this quick one before any comment comes in. Have you looked at the issue of coping styles or individual animal personality? Because there are some, if we are placed under the same condition, stressful condition, my response may be different from your response, and probably this can have effect on maybe the changes that you see in the hippocampus.
I wouldn't be surprised at all. We have not looked at it yet. We have not looked at it yet. I think it's a question of doing a separate experiment where we would have to first select the animals on their coping style, then expose them to stress, then see how that changes what in the response in the hippocampus. And we just haven't done that. I mean, as I said, every experiment takes a lot of time, so there's only so many we've been doing so far.
Yeah.
Good question, though. I think it's really interesting. Because if we can select for coping styles that deal better with chronic stress, that would be another way of reducing, welfare concerns by selecting animals that deal with it better.
Yeah, and can multiply them and
Exactly.
Yeah.
Although, of course, that might have other issues, right? Because if everybody's the same personality in a flock, there might be more fighting. There might be all kinds of, who knows? There's
life will be boring.
Yeah, exactly.
So, any comment from the participants? Please just comment. Feel free to talk.
Hello.
Hello, Dr. Rufus.
Yeah. Right. My name is Rufus Akinyemi. Thank you very much. I'm sorry to join a bit late. I was in the meeting with the provost of our medical school. I'm from the University of Ibadan, Neuroscience and Aging Research Unit. Thank you very much for a very elegant presentation. I wonder if you might have spoken about this before I joined it, so permit me if I'm asking about something you talked about before. Did you pick any significant cognitive deficits in the stressed birds, knowing that
we didn't
all right? Knowing that the hippocampus is very, very important.
Yes. So I mean, somebody's asked memory. We didn't test it. Essentially, we did.
From your research work and from your research group, thank you so much. We hope to have more of you anytime in the future when we have the opportunity. Thank you so much. I also want to appreciate all the participants from different parts of the world. Thank you for always being there, to support us and to make this group going on. Thank you so much. And with that, I say have a lovely day wherever you are. Thank you and bye-bye. Bye.